In this video, we're going to talk about capillary exchange. And so there are a number of processes that are involved in capillary exchange. First of all, diffusion, which involves the movement of solutes. Second, transcytosis, which involves the movement of specific solutes, and we'll get into the reason for the specificity later. And third, bulk flow, which is filtration and reabsorption. Now, bulk flow is involved in the regulation of the overall volume of the blood and the overall volume of interstitial fluid. Solutes will diffuse down their concentration gradients from blood into the interstitial fluid that surrounds them and from there into cells. So here are some of the solutes that actually diffuse. The first is oxygen, and I've got a picture of a red blood cell here, and oxygen from the lungs is going to diffuse down its concentration gradient from the lungs into the red blood cell. Once the red blood cell actually gets to the tissues, you're going to see the diffusion of oxygen from the red blood cells to the tissue cells. So oxygen is a solute and can diffuse down the concentration gradient from blood into the interstitial fluid. Glucose is also a solute that will diffuse down its concentration gradient from the blood into the interstitium. Same thing with amino acids and also with hormones. Likewise, carbon dioxide and wastes will diffuse from the higher concentration in cells into the interstitial fluid and then ultimately into the bloodstream. So carbon dioxide is probably the best example of this. It is produced, of course, as a product of cellular respiration and will diffuse out of cells into the interstitial fluid and from there into the bloodstream. The process is a little bit more complicated than that, but we'll cover the complications when we talk about the respiratory system. Now, proteins are actually too large to diffuse out of the bloodstream unless you're talking about sinusoid capillaries. So if you'll remember the three types of capillaries: continuous, fenestrated, and sinusoid. The fenestrated capillaries have openings, but they're too small for proteins basically to go through them. And so only the sinusoid capillaries with these really large openings, large intercellular clefts, and incomplete basement membranes, these large openings in the capillary walls of sinusoid capillaries will allow proteins to move in and out of these capillaries. So speaking of fenestrated capillaries, a greater variety of substances can move in and out of this capillary than they can in continuous capillaries. So I want you to remember that continuous capillaries have continuous cell membranes. There are no pores in the capillary. And that means in continuous capillaries, you don't see much of the same types of movement. When you move to fenestrated capillaries, there are a number of ways that substances can move in and out of the capillary. So let's zoom in on the bottom part of this diagram and take a look. Okay, so the first one that I'm going to mention on the left is basically diffusion through the membrane. Now, we've discussed diffusion through the membrane before. Basically, it's going to be small lipid-soluble substances generally. The second mechanism is movement through intracellular clefts. So what's going to happen here is that you're going to have water-soluble substances moving through here as opposed to lipid-soluble. So water is able to move between the cells here, the intracellular cleft, and so of course it will carry solutes with it. In fenestrated capillaries, you have an endothelial fenestration or pore in the cell membrane. And what's going to happen here is you can have movement through the fenestrations. These are going to be water-soluble substances, but these are going to be substances that might be a little bit too big to make it in between the cells in the intracellular clefts. Finally, we get to the fourth mechanism of transport, and that's transport via vesicles or calveolae. And so, if you'll recall, calveolae are actually invaginations of the cell membrane, and you're going to end up with a very small protein coated sort of miniature vesicle. So depending on the size or whether the cell is trying to bring something specific in, you can either have a transport vesicle, a pinocytotic vesicle, or calveolae. So transcytosis gets used by large and lipid-insoluble proteins. These cannot cross the capillary walls in any other way. So examples of this would be insulin, antibodies in the fetal circulation. So here's a picture of what that's going to look like. I want to orient you to this diagram real quick. So here we have an IgA secreting cell. So IgAs are immunoglobulins, they're antibodies. And so this cell, this IgA secreting cell, has just secreted an IgA, again a type of immunoglobulin. And so in order for this to work its way from that cell through or into another cell, through the cell, the very first thing that's going to happen is that it's going to bind to a receptor that is specific to that IgA. Okay, so here you see the receptor on the left. Just to the right of it, you see an IgA that is actually bound to that receptor. Now here's another point, that these cells have tight junctions, and there's no way for the IgAs to pass in between the cells in this case. This is the reason why you're going to want to use transcytosis. So you can basically see what happens. The IgA binds to the receptor, it gets brought into the cell, it gets transported through the cell, and then goes through exocytosis on the opposite end of the cell. So in this case, the IgA has passed completely through the cell, and this is transcytosis. So once again, insulin does this, and also antibodies in the fetal circulation will do this as well. Now we've talked about diffusion, we've talked about transcytosis, we've talked about basically passage of materials between cells. And so how do you get a large number of ions, large number of molecules, or molecules in a fluid moving in the same direction? This is going to be something that's called bulk flow. And if we zoom in on this portion of the diagram, this is something that happens in capillary beds. You see filtration occurring on the arterial side of the capillary bed. And so in all capillaries beds of the body, there's about 20 liters of fluid filtered out per day. Now this is almost 7 times the total plasma volume. And how would you get 7 times the total plasma volume filtered in one day? Well, there's also a lot of reabsorption, and we'll take a look at that in just a moment. So what I want you to see here is that we're on the arterial side of the bed, and that fluid is actually moving out from the capillaries into the interstitial space. So this is not diffusion. I want to make this clear. This is not diffusion. This is a movement from an area of higher pressure to an area of lower pressure. So what that means is that these materials, the ions, molecules, particles in a fluid, you're going to have a lot of different things moving in unison. If you'll recall, in diffusion, particle size can actually make a difference in the rate of diffusion. And so if you had a variety of different materials that were diffusing across a membrane, they would diffuse at different rates. In this case, with bulk flow, because it's based on pressure and not diffusion, you have a lot of different types of materials moving in unison from the capillary to the interstitial space. Finally, these will move at rates that are actually greater than in diffusion. And so I've mentioned that diffusion is a very fast process on the molecular level. Well, this is even faster. Now I've just defined filtration, and this is the pressure-driven movement of fluid and solutes from blood into the interstitial fluid. After this, after these materials move into the interstitial fluid, they will move into the cells surrounding the capillary. But it's not actually pressure-driven movement that does this. How do you generate the pressure that's used in filtration? Well, it's hydrostatic pressure, movement of fluid, and this pressure is actually generated by the ventricles. Most commonly the left ventricle, because it's involved in circulating blood around the systemic circuit as opposed to the pulmonary circuit. So again, the pressure that's generated by the heart, when you reach the capillaries, the plasma hydrostatic pressure is usually about 35 millibars. At this point, the interstitial fluid hydrostatic pressure is 0 millibars. As we learned with the cardiac cycle, fluid is going to move from higher to lower pressures. And this is the reason why fluid will move from the capillaries into the interstitial fluid. The opposite number of filtration is going to be reabsorption. And so this is pressure-driven movement of fluid and solutes from interstitial fluid back into the bloodstream. So whenever you talk about filtration, you're talking about things moving out of the bloodstream. Whenever you talk about reabsorption, you're talking about materials being reabsorbed back into the bloodstream. So anytime you hear filtration, think exiting the bloodstream. Anytime you hear reabsorption, remember re-entering the bloodstream. So if we take a closer look at this, now remember in the last slide, I told you that 20 liters of fluid per day gets filtered. Of those 20 liters, only 17 liters actually gets reabsorbed into the capillaries at the venous end. And so where do the remaining 3 liters of fluid a day, where do they go? Well, about 3 liters a day actually get removed by the lymphatic system, which we're going to see a little bit later on this semester. Now, what type of pressure drives filtration? It's not hydrostatic pressure, because hydrostatic pressure comes from the ventricles. That's always going to push blood out of the capillaries. What is it that pulls blood back into the capillaries? This is actually going to be osmotic pressure. One of the things that we covered in lab in AMP1 was osmosis. And if you recall, I told you that osmosis actually has sort of a force of its own. And so osmosis is actually going to be the force that brings fluid back into the capillaries. Now, there's a type of osmotic pressure called oncotic pressure. And oncotic pressure gets produced by plasma proteins that are in the bloodstream. We just determined that proteins are too large to leave capillaries, with the exception of sinusoid capillaries. Now, sinusoid capillaries are really rare, and continuous capillaries are much more common. And what this means is that in either continuous or fenestrated capillaries, proteins are not going to make it out of the bloodstream. One thing that usually gives students a little bit of trouble here is to say, well, what do proteins actually have to do with osmosis? Because much of the time when we've talked about osmosis prior to now, we've talked about the solutes causing osmosis to be salt and maybe glucose. Those certainly were the ones that we covered in lab, were salt and glucose. However, anything that is dissolved into the bloodstream as particles can actually act as an osmotic solute. And so what that means is you've got a lot of protein molecules present in the bloodstream that are not present in the interstitial fluid. And that means you have a higher concentration of these protein solutes in the capillaries than you have in the interstitial fluid surrounding it. And so what that means is that water is going to go from an area of lower to higher solutes, and in this case is going to be drawn in towards the blood plasma. Now, the combination of hydrostatic pressure and oncotic pressure results in a measure called net filtration pressure. So net filtration pressure, as you can see from the word net, it means that something is going to add to this value and something is going to subtract from this value. Now, hydrostatic pressure causes filtration and gets defined as a positive value. Oncotic pressure causes reabsorption and gets defined as a negative value. So if your net filtration pressure is positive, that means that hydrostatic pressure is greater than oncotic pressure, and therefore fluid moves out of capillaries. If the oncotic pressure exceeds the hydrostatic pressure and you have a resulting negative value, this means that fluid is going to move into capillaries. And so are conditions going to be the same all the way across the capillary bed? The answer to that is actually no. So let's get oriented to this particular figure. The first thing that I want to point out is that there's an arteriole on the left and there's a venule on the right. And so basically, when you pass from an arteriole to a venule, you're going to divide up into capillary beds. two things are going to happen as you move from an arteriole into a capillary bed. First of all, you're going to have extensive branching. That's going to increase the cross-sectional area of the vessels, and you're going to drop in pressure and you're going to drop in velocity of the blood. The other thing that's going to happen is just basically resistance as you continue to move through the bloodstream. So resistance and an increase in cross-sectional area means that you're going to have, from the arterial end into the capillary bed, you're going to actually have a loss of hydrostatic pressure. So what we're going to do is we're going to compare the net filtration pressure on the arterial side, on the arteriole side, and on the venule side. And so what we'll take a look at first is actually the net filtration pressure on the arteriole end. So the area that I just expanded shows you net HP minus net OP. So this is going to be hydrostatic pressure minus osmotic pressure. Now, you've got to compare some things here. So what's the hydrostatic pressure in the capillary versus the hydrostatic pressure in the interstitial space? Well, the hydrostatic pressure inside the capillary is 35 millibars. This is the pressure that we referred to as being caused by the left ventricle. The hydrostatic pressure in the interstitial fluid is 0. And the reason why it is 0 is because there's no pump out in the interstitial fluid at all. So there's no structure there to generate hydrostatic pressure. So our net hydrostatic pressure will be 35 millibars in the capillary, 0 millibars outside the capillary. So here you've got a force of 35 that is pushing out. When we talk about osmotic pressure, there are a lot of plasma proteins in the capillary that have not been pushed out along with other solutes. So they're going to exert an osmotic pressure at about 26 millibars. Because very few proteins have left the capillary, there are very few proteins in the interstitial space. There are a few. So they're going to exert an osmotic pressure of 1 millibar. So 26 minus 1 means that our osmotic pressure in the capillary is 26. The osmotic pressure in the interstitial fluid is 1. And that means that the osmotic pressure will try to pull fluid back into the capillary at a force of 26. Osmotic pressure on the outside tries to pull it out at a pressure of 1 millibar. So your net osmotic pressure is actually 25 millibars. Now, one thing we established in a previous slide is that filtration pushes out and osmotic pulls in. So you have a net hydrostatic pressure pushing out at 35 and a net osmotic pressure pulling back in at 25. And so that means you have to subtract the 25 value from the 35 value. That gives you a net filtration pressure of 10 millibars. And that means that at 10 millibars net filtration pressure, since it's a positive value, fluid is going to move out on the arterial side of the capillary bed. Now let's consider the venule end. And I hope you think it's clever that I put this in blue for the venule. So let's take a look at our values here. So by the time you reach the venule portion of the capillary bed, the hydrostatic pressure is less than it was in the arterial end, again because of resistance and because of capillary branching. Now, one thing to say is that you do see capillaries sort of joining back together at the venule side of the capillary bed. So you're going to have a little bit of an increase in the pressure that's present in the capillary. So in this case, our net hydrostatic pressure is 17 in the capillary, no longer 35. It's been cut by about 50%. So you have a net hydrostatic pressure of 17 because your hydrostatic pressure in the capillary is 17. As we established before, there's no hydrostatic pressure in the interstitial fluid. So you have a net hydrostatic pressure of 17. Now, if we talk about osmotic pressure, it's going to be the same as it was on the arterial end of the capillary bed because the number of proteins in the capillary has not changed. The number of proteins in the interstitial fluid has not changed. So you have the same values. 26 millibars osmotic pressure pulling in, 1 millibar osmotic pressure pulling out. And this means our net osmotic pressure is once again going to be 25 millibars. Now what makes a difference here is that the hydrostatic pressure has been reduced from 35 to 17. So instead of going 35 minus 25 and getting a net filtration pressure of 10, in this case you take 17, subtract 25 from that, and your net filtration pressure is negative 8. So what this means is that at a negative value, fluid is going to move from the interstitial fluid back into the capillary beds. So earlier we mentioned that you have about 20 liters of fluid that are filtered per day through capillary beds and about 17 liters of fluid that is reabsorbed back into capillary beds. So why is there a difference, 20 versus 17? This is the reason why right here, because your net filtration pressure at the arterial end is 10 out. Your net osmotic pressure at the venual end, your net filtration pressure at the venual end is negative 8. 10 out, 8 in. It means that overall in the capillary bed, you've got some fluid that's been pushed out on the arterial end that doesn't get reclaimed at the venual end. This is the reason why we have to have lymphatic vessels to pick up the rest of the fluid. This is another example or another diagram for bulk flow, and I want to make it very clear that this is only for the arterial side or at the very beginning of the capillary bed. So close to the arterioles you have a hydrostatic pressure of 35 out. You have a hydrostatic pressure of 0 in. So again, there is no hydrostatic pressure in the interstitial fluid to push the fluid into the capillary. So our net hydrostatic pressure is 35. In terms of osmotic pressure, at the very beginning of the capillary bed, your plasma proteins, which are in the capillary, pull fluid out of the interstitial fluid into the capillary at a force of 26. Now there are a very few proteins that are out in the interstitial fluid, and those few proteins that are in the interstitial fluid will pull fluid out of the capillary. But if you'll notice, the osmotic pressure into the capillary is 26. The osmotic pressure out is 1. And so here you can see this. Hydrostatic pressure 35 out, 0 in, and this is going to be 35 out overall. Osmotic pressure 26 in, 1 out, 25 overall. So if you have 35 overall out, 25 overall in, it means that your net filtration pressure will result in 10 millibars of pressure moving overall fluid from the capillary into the interstitial fluid. This brings up something that's called Starling's law of the capillaries. So this law states that the volume of fluid in solutes that gets reabsorbed is almost as large as the volume filtered. It also states that the small portion that you don't get reabsorbed back into the blood will be removed by lymphatic vessels. So this is just another diagram of the capillaries down below Starling's law here. So at the arterial end, your net filtration pressure is 10 millibars. Again, 35 greater than 25. At the middle of the capillary bed, your net filtration pressure is going to be 0, because as you've moved from the arterial end to the mid capillary, your hydrostatic pressure overall has dropped from 35 to 25. So at mid capillary, 25 in, 25 out, your net filtration pressure is 0. So there's no net movement. Now once you get to the venous end, after you've had a further drop in hydrostatic pressure, so your capillary hydrostatic pressure is going to be 17, your osmotic pressure remains consistent at 25, and this means your net filtration pressure is negative. That's going to indicate reabsorption. But again, as part of the big picture here, if you realize that your net filtration pressure is 10 millibars out at the arterial end and only 8 back in at the venous end, there's going to be some excess fluid, and again, that is what the lymphatic system will actually pick up and do, to reabsorb the fluid that is excess of filtration over reabsorption. So edema or swelling due to fluid, this is usually not detectable until your interstitial volume is about 30% higher than normal. And so edema can happen if filtration exceeds reabsorption. And this can happen in one of two ways. First of all, there can be excess filtration in which there's a higher hydrostatic pressure than normal. And so increased capillary blood pressure. This is the reason why you can see some swelling in the extremities of people with hypertension. It's because their capillary hydrostatic pressure is so high that the net filtration pressure is greater than 10. Once it goes to about 30% above normal, then there will be so much filtration. There will be so much more filtration than there is reabsorption that the lymphatic system can't handle the overload. So excess filtration can happen as a result of increased blood pressure, but it can also happen due to increased permeability of the capillaries. And so this happens in inflammation, where the production of histamine and heparin cause the capillaries themselves to become a great deal more leaky. And so if the capillaries are leaky, then even a standard net filtration pressure on the arterial end will push so much fluid out that the lymphatic system can't handle the overload. Now, the other main mechanism of filtration exceeding reabsorption is when you have inadequate reabsorption. And so this might be some sort of problem with the lymphatic system. And so a couple of things can contribute to this. One is decreased plasma proteins. And with decreased plasma proteins, this can be, I think the most classic example of this happening is in-state starvation, in which the body begins to actually break down blood proteins to serve as a protein source. So if there are fewer proteins present in the bloodstream, then you won't have as much osmotic pressure, and your net filtration pressure at the venous end will actually be might be -5, might be -6, but it will be less than -8. And so in this case, a normal filtration greatly exceeds the inadequate reabsorption, and you get swelling because of the decreased number of plasma proteins. Another thing that can occur to cause decreased plasma proteins can be liver disease. I mentioned malnutrition. I mentioned endpoint starvation. That's going to be malnutrition, burns, and kidney disease. Another thing that's not mentioned here that can cause inadequate reabsorption is actually parasitic infections. And so there's a disease called elephantiasis that can result in localized edema. And if I had to guess, I would guess that these pictures are pictures of elephantiasis, or someone with a parasitic infection. Now, in that case, the lymphatic vessels are actually physically blocked by the parasites, and that causes inadequate reabsorption, even with a normal filtration level.